Alright, this video is going to start a series of videos on what is called microbial genetics. I'm going to click on the overview. This is a pretty complicated subject. You get to learn a little bit about coding. You get to learn about genetic code. Now, this is an outline that has been used in our class before. It's got a lot of information on it. I try to give you as much as possible. You really don't have to buy the textbook if you don't want to, of course. But here is a nice introduction, and then also there is a PowerPoint once again that was used at one point in class. Kind of been updated a little bit, but this has also got some really good pictures, some good information. Yes, thank you. Okay, some good pictures, just going through everything. Now, this one here, it says the microgenetics video outline. This is a video that goes through every part of this outline, which is the same as the others. It's just it shows you how to do everything. It goes through the parts of DNA, genotype, phenotype, and then what is transcription, translation, genetic code, and here is the codon chart. And here is the video PowerPoint. The video kind of references this right here. Goes through all of this, and then also this codon chart. It's also in the outline, but if you wanted to have this separate for whatever reason, oops, and I didn't mean to make it that big. I think that's twice it's happened. And you'll get the gist of what this is. These are the codes for how the transfer RNAs know where to attach. This is phenylalanine, leucine, things like that. Okay, so I guess let's go ahead and get started with our, let me go back to this. Let's go ahead and get started with our breakdown of microbial genetics. All right, we are going to start our section on DNA. When you take a biology course, this is standard procedure. You cover DNA and genetics. This is actually one of the more interesting things you can cover. You really can apply this to real life. So we're going to start here first, and then I do have some videos to show you within these videos. Some sections from movies. Hopefully they won't get banned from YouTube. But we'll start here first. Remember, deoxyribonucleic acid. That's found in the nucleus of all cells, and even bacterial chromosomes have DNA. So back to our cell model from a couple of weeks ago. So the DNA is housed inside the nucleus. So remember, when cells divide, they duplicate the DNA. You end up with what are called sister chromatids. You have double the amount of DNA if you're going to make a new cell. Well, when a cell is going to make something, make a protein, anything, skin pigment, sweat, anything a cell is going to produce, it will copy a section of DNA in the form of RNA, and then the cell, and specifically at the ribosome, and we'll go to those in a minute, will then read that genetic code, and then the cell will produce whatever the gene is telling the cell to produce. Here's a DNA molecule. Remember, DNA is double-stranded, and it has base pairs. The C, the G, the T, and the A. Remember, this is cytosine, guanine, thymine, and adenine. And that's normal base pairing. A's bind with T's, and vice versa. You can see there. C's always bind with G's, and vice versa. That's normal base pairing. Now, these two guys, James Watson and Francis Crick, I'll just show them. They're the ones that came up with how the DNA molecule is constructed. And this is their famous model. My understanding, that's actually in Belgium, this model is, but you can see them pointing to it. But they came up with the theory about DNA and the functionality. And, prior to these guys, there wasn't very much known about genetics. And if you remember also, DNA was made of what are called a nucleotide. It was a phosphate, a pentose sugar, and what is called a nitrogenous base, which is what the G's and the C's and the A's and T's, that's what they represent, the base pairs. So there are categories and it depends on their actual biological or biochemical makeup. Purines and pyrimidines is what you hear about these. The two carbon bases, adenine, guanine, are purines. It's just a category. And then the one carbon nitrogenous ring bases, thymine, uracil, and cytosine, are pyrimidines. Now, thymine and uracil are practically the same. Uracil is what is part of RNA. Cells actually synthesize RNA from a DNA template. And thymine costs a lot, scientific term, metabolically to make. So instead of making thymine in RNA, cells will produce uracil, which costs less metabolically. I'm going to leave it there. We're going to be very general with all this. All right, DNA replication. I just mentioned this. There are times where DNA has to be copied or doubled. This diagram here is actually showing that. You see it splitting, that this happens all the time. This is how genes are copied. And what ends up happening is when the DNA splits like this, there are enzymes that do this, there will be a complementary strand made, a copy of a gene, and then that gene is actually copied into a strand of messenger RNA. So we've kind of talked about that. Cells will double their numbers as far as chromosomes, double organelles before they divide, and they also do copy genes as well. So in your PowerPoint I gave you, this was actually a really good, this kind of showed along it. See, DNA is double-stranded, it's a helix, but you can see here gene 1, gene 2, gene 3, and there it would be showing a part of that really blown up large, but genes constitute what are called traits, and everybody has a genotype, that's the genetic code, and then a phenotype is the expression of that code or the traits that are produced as a result of having that genetic code. Give you an example. If you're type A blood, if you're A negative, that means the genes you got from that came from your parents. You have the genetic code. You have an A antigen or an O antigen for blood typing or blood types, or you have two A's. And that's what makes you type A blood. Those are genes. The plus and negative is called the Rh factor. If you're A negative, you do not have a antigen D, which is also that comes with a gene. They're called alleles. So that would make you Rh negative. So on this particular diagram, remember these are histones, so it's unraveling the DNA, but all that's found within the nucleus. All right, we're about to get to our first little video within the video. It says a typical gene has thousands of nucleotides, that's true. And then a single DNA molecule may contain thousands of genes. The phenotype are the physical traits that arise from having that genetic code. And we're going to get to this in a minute. All right, so starting off first, we're going to do this scene from Jurassic Park. Explains it great between genotype and phenotype. And then I'll throw this again in later. But here's that scene from Jurassic Park.
Here he comes. Well, here I come. Hello. Hello. Say hello.
Hello.
Hello, John. Oh, yes. I've got lambs.
Well, fine, I guess. But how did I get here?
Well, let me show you. First, I'll need a drop of blood. Your blood.
Right. Oh, John, that hurts.
Relax, John. It's all part of the miracle of cloning.
Hello, John. Hello, John. Hello.
Hello, John. From what?
Hello, John.
Flow extractions never recreated an intact DNA strand.
Not without master sequence codes.
Good afternoon, John.
DNA from what source? Where do you get 100-million-year-old dinosaur blood?
Shh.
What? Oh, Mr. DNA, where did you come from?
From your blood. Just one drop of your blood contains billions of strands of DNA.
the building blocks of life. A DNA strand, like me, is a blueprint for building a living thing. And sometimes animals that went extinct millions of years ago, like dinosaurs, left their blueprints behind for us to find. We just had to know where to look. 100 million years ago, there were mosquitoes, just like today. And just like today, they fed on the blood of animals, even dinosaurs. Sometimes, after biting a dinosaur, the mosquito would land on the branch of a tree and get stuck in the sap. After a long time, the tree sap would get hard and become fossilized, just like a dinosaur bone, preserving the mosquito inside. This fossilized tree sap, which we call amber, waited for millions of years with the mosquito inside, until Jurassic Park scientists came along. Using sophisticated techniques, they extract the preserved blood from the mosquito and, bingo, dino DNA. A full DNA strand contains 3 billion genetic codes. If we looked at screens like these once a second for 8 hours a day, it'd take 2 years to look at the entire DNA strand. It's that long. So, it's full of holes. Now that's where our geneticists take over. Thinking machine supercomputers and gene sequencers break down the strand in minutes. And virtual reality displays show our geneticists the gaps in the DNA sequence. We use the complete DNA of a frog to fill in the holes and complete the code. And now we can make a baby dinosaur.
This scar is only temporary.
All right. Well, I think everybody, that movie's getting old. It did a really good, even though it's a little cheesy, but it did explain what DNA is and the whole building of a dinosaur. We're going to cover that in this section. How do you start out with a genetic code, and then how do you basically build something from that? So I will start here next.